Hi there and welcome to another of the ASH PE screencasts. And this is the third in the series that's linked to the energy system. And the system that we're going to be looking at today is the anaerobic lactic acid energy system, or the glycolytic system. So let's get started. Let's just recap where we are and what systems we've covered very quickly just in this one slide. So we have our adenosine triphosphate initially. We have the exothermic reaction, hopefully you can remember that, and that's the breaking away of the phosphates from the adenosine. And that gives us our energy, but it only lasts for about 2 to 3 seconds. So the resynthesis of the adenosine diphosphate is first of all from our phosphate creatine system. And this is an endothermic reaction where the phosphate is returning to the adenosine diphosphate. And there we have it, back in its full entirety. Therefore, we're able to produce that energy again. But the combination of the phosphate creatine and the ATP system only gives us about 8 to 10 seconds worth of high-tense exercise. So we obviously need some more. So here we are. We have our adenosine diphosphate plus a phosphate. We have the creatine kinase, which is the enzyme that's present that stimulates the breaking up of the phosphate creatine, but there is no more phosphate creatine in our muscle stores. So we then have to turn to a new system, and that's the one we're talking about in this session here, which is the lactic acid energy system. Okay, so the lactic acid energy process, and also known as the glycolytic system, starts when we have a buildup of adenosine diphosphate. So once this happens, it triggers an enzyme. Now, the enzyme is known as phosphofructokinase. And what this does, it begins the process of the breakdown of the next available food source, which is glucose. Okay, so glucose is an energy-rich molecule, and it's circulating in the bloodstream. So if the glucose levels drop, then glycogen phosphorylase, another word for that is GPP, an enzyme which then starts the breakdown of the stored glycogen in the muscles and the liver. This one is released as well, so it's very important we note these enzymes. And it converts the glycogen into glucose to maintain its concentration in the bloodstream. So that will be from the liver and also from the working muscles as well. Now, glucose is easily accessible because of this fuel being broken down, and then we can use this to allow us to have the continued ATP resynthesis. And the location for it is in the sarcoplasm. So if you have a look on this diagram here, this is our sarcoplasm right here, which is this the pink component there, as opposed to the dark red, which is the, if I believe they're the myofibrils. And then the yellowy ones there are the muscle fibers. So it's right, it's present where we need it, and that's really important to note as well. So what we've got is what's happening and then where it's happening. All right, so let's move on. So the breakdown of the glucose in the absence of oxygen is what we've got, and the issue that we have there is this is now called anaerobic glycolysis. And obviously we would remember that from our GCSE that the anaerobic is without air. So what happens then is we have pyruvic acid formed. So it does actually allow us to resynthesize, but it's important to note this, that once we've gone from our adenosine diphosphate to the adenosine triphosphate, we need to work out, or we need to know, how many moles are available, which is a measure or a unit when we're talking about these components here, from this pyruvic acid. So for every glycogen mole that is broken down, it actually gives us 2 adenosine triphosphate moles. All right, so there we've got the ratio 2 to 1. So it's okay. It's not the most efficient way that we can actually get adenosine triphosphate resynthesized. However, there are some other advantages to it. What it does allow us to do is work at a very high intensity. So, for example, a 400-meter run, something like that, or a 200-meter sprint. And usually we can say that it'll last for about 3 minutes. Now that in itself, you think, well, great, that I can work at absolute max for 3 minutes. That's not entirely true. Ordinarily, we would say, super high intense exercise can be carried out for 1 minute. But then what we'll do is then lower slowly down to that 3 minute point when we will then have the onset of blood lactic acid. But we'll talk about that in a minute. So time-wise, what's happening is we've got high-intensity exercise for at least 1 minute, and then it tapers off for the remaining 2. Okay, let's move on. So what we've got happening is, another term for it is glycolytic adenosine triphosphate resynthesis. Okay. So with this system, O2 is not available to support this resynthesis because it's anaerobic, as we mentioned before. The intensity is way too high. So we're not going to be able to last forever. And there are other side effects, as I'm sure you know. So with the element of pyruvic acid, what actually happens is another enzyme is released, and this is the lactate dehydrogenase, or LDH enzyme. Now this is released because of this resynthesis, but it's anaerobic. So because there's no oxygen there, the lactate dehydrogenase, LDH, is released, and therefore this is the catalyst for the conversion of pyruvic acid into what you and I know, which is lactic acid. Now, what's the knock-on effect of lactic acid? I'm sure a lot of you have had this experience. But what it's actually doing is slowing down the adenosine triphosphate resynthesis. It doesn't allow it to take place as quickly, so therefore your performance is going to slow down. It can't continue that process. As more lactic acid builds up, what you're going to have is the onset of blood lactic acid, and that's when it moves past a certain threshold. And I think the measure is a 4. Yes, I'm pretty sure it is, but look, don't quote me on that for now. But let's go back through this then. So we have the glycolytic ATP resynthesis, that's okay. But the knock-on effect of that is pyruvic acid, because it's it's anaerobic, and this stimulates the enzyme lactate dehydrogenase, and that is the catalyst for the conversion of pyruvic into lactic acid. This slows the ATP resynthesis, and therefore we have the onset of blood lactic acid. Okay, so let's look at that in just a graph as a total. So we started off with adenosine diphosphate. Then we've moved into phosphofructokinase, and this was because of the increase in the amount of the adenosine diphosphate there. From that, we've then got glycogen phosphorylase, which is this one here, and this is because of the reduction in the amount of glucose within the blood. And so what it does is stimulates the conversion of glycogen into glucose, which, as a knock-on effect, yes, it does give us pyruvic acid. But in turn for that, we do get 2 phosphates, which allows us to have ATP resynthesis. And remember, we said it's a ratio of 2 to 1. So 2 ATP for one of the glycogen molecules that are broken down. However, the side effect of working without oxygen, so anaerobically, is that we have the LDH, which is the lactate dehydrogenase, is released. And this then in turn gives us our lactic acid. The knock-on effect of that is a slowing down of the enzyme's ability to break down each of the glycogen stores. Okay, so that's what it's doing. Now, the other thing that we would need to know, especially for an exam question, I think this is a really, really important table, these next two, because it tells us exactly what we need to know about each one of the different systems. So, the type of reaction, that's what we need to know first of all. So try and go through it as a process. It's an anaerobic process. What's the site of the reaction is at the sarcoplasm. Very, very specific, that. So what's the food fuel that's used? It's glycogen or glucose. And the controlling enzymes. Now, what you can do, and they're happy for you to do this in your exam answers as well, is note them down in the annotation format like that. So GPP, phosphofructokinase, and the lactate dehydrogenase. It's absolutely fine. Also, note what the yield is. So what are you actually getting from the system? And use the term mole. So 1 mole glycogen yields 2 moles of ATP. And then in this specific stages, there are three real stages is the anaerobic glycolysis. So what's happening with the glycogen first and foremost? So glycogen is broken down into glucose, which gives us pyruvic acid plus the energy. So that's very important. And then the second component to this is the lactic pathway. So the pyruvic acid, we know that then turns into lactic acid. And what we can do then is just note what happens in relation to the energy. So we have energy plus 2 phosphates. So that's the 2 phosphates plus the 2 adenosine diphosphates. And that gives us 2 adenosine triphosphates. And that is where the endothermic reaction has actually taken place. The knock-on effect, as we all know, is lactic acid, or byproduct is the term that we can use. And it's up to about 3 minutes depending on the intensity. All right, and then let's look at the strengths of this system and also the weaknesses. What it's really, really good for is having no delay for, you haven't got to wait for oxygen or large fuel stores to resynthesize. So in that sense, it's really good. It's relatively fast to break down for ATP resynthesis because it's at the absolute location. It's a perfect location, isn't it, in the sarcomere. And then provides energy for high intense exercise, which is really, really good. And oxygen's not obviously being able to do that, so that's an advantage. Knock-on effects, though, are it's fatiguing byproduct of lactic acid, which reduces the enzyme activity, and therefore we can't resynthesize as quickly. And it's a relatively low yield. So if you think about it, it's only 2 to 1. So through all of that, and also the lactic acid production that we've got there, is it's only 2 ATP. So we're going to need to look for something pretty quickly, aren't we? And also, if you get the onset of blood lactic acid, it can be quite a lengthy recovery. All right, so what we've got there is an overview of the process. Do pause the screencast, go back through the process until you're really clear on it. Have a look at the table back here. I think it's really important, the types of reactions and all the key components to a really good exam answer. And then last but not least is if we're looking at the strengths and weaknesses. That way we could answer any comparative questions, what I think would be really important. Okay, so there's your summary. So you can pause that at any point, have a look at that. Do go back over it. I think they're the 3 main sections that we've looked at. And that's the end. Okay, so thanks very much, and I'll speak to you soon.